I am Albert Higgins Chen. I am Raghav Sehgal's mentor. Um, the graduation of a student is one of the proudest moments for a mentor. Raghav and I, Raghav and I first met when we were both members of Morgan Levine's lab. I was a postdoc, he was a grad student. And when Morgan joined industry, I started my lab, and Raghav joined my lab, along with Yarrow and John, who will both be defending later this year. John next week. The lab studies aging and longevity, and as you'll see today, Raghav has done a ton of cutting edge work in this space. But before we delve into that, it's worth pointing out that he's done a ton for the field outside of the lab. He's a social media maven on Twitter, X, TEDx, numerous podcasts and conferences. He is a principal at the Longevity Tech Fund venture capital firm and the director of bioinformatics for the Associated Healthy Longevity Clinic. His work has been commercialized as Symphony Age by our amazing collaborators at True Diagnostic. And for all of this, he has represented the longevity field as part of Forbes 30 Under 30. And I'll let this video. But in the lab, he's kind, he's curious, and he's insanely fun to work with. Importantly, he's there for others. And one of my top priorities in starting the lab was to create a culture of collaboration and caring. And I could not have done this without Raghav. He was always there to help others, and he would let me know when others needed help. And about a year ago, I recognized he was ready to take on a larger leadership role within the lab. And so he was central to this group collaborative project, which he'll talk to you about today, now termed Translate. He organized together with Daniel these coding weeks. As you'll see in his talk, the scale of this project required everyone in the lab to work together. And to amp everyone up, he created these. This is literally the slides from his presentation. He wanted to amp everyone up. And it's bigger, it's better, with more data sets, more clocks, more data, more clocks. And he told these stories of science to motivate everyone, to get everyone excited. For example, Raghav Sehgal, who discovered rapamycin, which extends lifespan and slows aging in mice. And Raghav used this story to remind everyone that science is hard. But if we keep on going, we never know where we might end up, and we may just end up changing the world. So Raghav, I'm proud of you. I'm proud of you as my student. I'm proud to call you my colleague and my friend. I'm trying not to cry.
Thank you, Albert. Thank you so much for that. Coming from you. I'm gonna try and move to my presentation, which give me a minute. It's gonna be some challenge. Okay. everyone see this? Yeah? Okay. Um, very honored to be here today. It's been a really long journey. And really thankful for that introduction, Albert. Thank you. But today is, as much as it's about sort of all of this, but today is actually about the science that we've been doing for the last 4 or 5 years, which not only that I've been doing, but a lot of people in the lab have been doing, that Albert, Morgan, all of these folks have been helping me out with, and that is what I'm here to share with you guys. So let's start. So my thesis is about translating geroscience to clinic. And it's titled Translating Geroscience to Clinic Using Machine Learning for Insightful Aging and for insightful and responsive aging biomarkers. And sorry. So how many of you have actually heard the term geroscience? And my lab members don't count. Neither do you, Joe. Well, some of you might have, some of you might not have, but actually the term geroscience or some usage of it has already been there in sort of in headlines, in newspapers, in our day-to-day lives for a very long time. So for example, NPR, probably the only news source we can trust nowadays, talked about the TAME trial, which is one of around metformin, a very common diabetic drug that's given to diabetes patients, and which may potentially prolong health span and lifespan. Or the Kardashians trying to measure their biological age. Check out season 4 finale. Shockingly, Khloe Kardashian was the youngest. I'm very shocked about that. Or this guy. How many of you know this guy? Hands up, yeah? Brian Johnson, the centimillionaire who's trying to live forever. You see, the talks about geroscience have been there around us in our culture for a very long time, but what is geroscience? So let's try to answer this question, and why is it really important? Well, I really like this definition. This definition was in a publication which had a couple of really famous authors, such as Brian Kennedy, as well as Luigi Ferrucci, who's the current director of NIA. And the definition goes something like this. Geroscience is a field of biomedical research which seeks to understand how aging processes drive chronic diseases in order to develop interventions to extend human health span and prevent disease onset with age. In simple terms, what it really tries to do, it's really trying to say is it's the study of biology of aging that can essentially extend human health span by building new longevity interventions and prevent the onset of chronic diseases. But why is this really important? Well, the simple reason is because age is the leading risk factor for a lot of chronic diseases. So actually on the y-axis, this is a log scale. And what you would see is these chronic diseases like cardiovascular disease, diabetes, stroke, dementia, all of these diseases tend to sort of increase exponentially as we age. Actually, the risk of developing any of these chronic diseases doubles every 8 years, which is quite bad. And actually, it becomes worse if I give you this next statistic. So 1 in 6 people will be over the age of 65 by the time I turn 65, and 1 in 4 will be over the age of 65 by 2100. By the time I would be probably 105, which is pretty bad. I don't know if I want to live with that. But more importantly, US will see a dramatic shift. There is, as I said, a more derogatory term called the silver tsunami, where we will see a dramatic shift in sort of baby boomers turning from sort of the middle age to being being dramatically older in their 60s to 85 age range. And this will potentially be a huge weight on our economic and social systems. Take, for example, our Medicare system. This will affect our economies. For example, the Medicare care system itself will go from being costing about 4% of the US GDP to costing about 6% of the US GDP. And a large part of the cost increase would be in physician services. What that means is that these people that are growing old significantly under the Medicare system will have to be cared for the chronic diseases that they're getting. Now, what do we do to really resolve this problem? Well, here I want to introduce the idea of lifespan and sort of health span. So essentially, lifespan is the total life you live, the total time you live. Often a lot of that, a part of that life actually is plagued with chronic diseases, especially your last few years. And so the first time you get a chronic disease is the time when is the end of what we call your health span. So the time from your birth to the hit of your first chronic disease is what we call your health span. Our current paradigm of medicine has really allowed us to extend our lifespan. So for example, what happens is we have been able to, for example, treat cancer, but we only treat cancer once we get it. Or we have been able to treat cardiovascular disease, but we only treat cardiovascular disease once we get it. We do not treat these diseases before the onset of them at all, right? And that's kind of the paradigm we have been working with in the context of medicine. And so what that has allowed us to do is essentially extend lifespan without really extending health span itself. And you can see this from this graph altogether. So this graph is really interesting in my opinion, because what it's really showing is that over the period of time, on the top, what you're seeing is that lifespan of individuals across the US has been increasing from 2000s to about 2020. But the health span, the onset of their first disease, hasn't quite increased. Rather, if you look at the difference between their lifespan and their health span, that has been increasing. So that means that the time that they've been living with chronic diseases has been increasing over these last 20 years. And which is not great, because not only are they in pain longer, are they sort of suffering for longer, they are also that is also increasing their healthcare costs, that is a larger burden on the economy, and they're probably not even able to sort of contribute as much in a lot of ways. And that, to me, is a huge problem and will only increase as we move further into the next few years. So what do we do about this? And to this, geroscience can potentially be a solution. So geroscience can be a solution in two ways. The first way it can be a solution is we can potentially build novel therapeutics, gerotherapeutics through clinical trials that can prevent the onset of these sort of chronic diseases by sort of prolonging sort of this health span. And the second thing that could happen is we could also sort of integrate sort of this preventative sort of gerocentric medicine model into our clinical practice. But to do this, what we really need to do is a way to somehow quantify aging itself. And for both of these cases, as I said, we need to quantify aging because of this one quote I really feel is really powerful from sort of the famous physicist, Lord Kelvin. What is not defined cannot be measured. What is not measured cannot be improved. And what is not improved is always degraded. And this is very true in the context of aging, if you think about it. If we cannot quantify aging itself, we will not be able to improve upon it. And we will not be able to build gerotherapeutics. We will not be able to treat it. And you might say, Raghav, we have such a good measure of aging, that is our chronological age, the number of candles on your last birthday, right? And in a lot of ways, that is used, right, in clinical practice. Many times, there are cutoffs for giving certain medications to individuals in clinical practice, or performing certain operations on individuals in clinical practice. But the thing about chronological age is it is not modifiable. If you have a certain gerotherapeutic or a sort of longevity intervention, it doesn't really go down, right? You cannot modify it, right? Or it doesn't vary based on how a certain person is aging, right? Because, for example, I'm not the healthiest person. I don't do a lot of exercise. On the other hand, my mentor, he is very, very fit, goes to the gym every afternoon. His chronological age should be potentially younger than mine, but he's not. So there should be a better way to sort of quantify these things. And that is where the idea of biological age comes in. which is potentially a measure of lifespan and health span. So what is biological age? So biological age is essentially a measure that can help us quantify sort of health span and lifespan. So in the most simplest terms, one can say that if your biological age is higher than your chronological age, you might have a potentially shorter health span and shorter lifespan. And if your biological age is lesser than your chronological age, you might have a longer health span and longer lifespan. And that's a very simplified definition. Of course, there are more complicated ways to define lifespan and health span and biological age as well. And this is essentially what an aging biomarkers try to quantify. So they tend to indicate sort of biological aging and sort of predict sort of these health outcomes in some ways. And there are different ways to be able to do this. We could have different types of omic data. We could have phenomic data, like MRI data, like brain MRIs to do this. We could have clinical biomarkers, sort of metabolic panels, immune panels, so on and so forth. We could have proteomic data that could do this. There have been proteomic clocks. But the most popular of them have been DNA methylation clocks. And actually, probably the creator of the DNA methylation clocks, Steve Horvath, is actually probably on this call right now. Pretty exciting. And what they really do is these DNA methylation clocks tend to predict sort of some kind of aging outcome. It could be chronological age, pace of aging, mortality, or it could be something more different, something like a physiological outcome like frailty, something that Janelle in our lab is working on. But what is DNA methylation? Let's talk about that for a minute. Why is this such an interesting sort of omic data type? So DNA methylation at the very core of it is, in my opinion, kind of the recipe of the code of life. So it's an interesting way of thinking about it, and sort of bear with me for a minute. So all the cells in our body have the same genetic material. They have all the same genes. So your brain has the same, your brain cell, your neurons have the same gene as your skin cell, right? But how does a brain cell, a neuron, know how to be different from a skin cell, right? It's through sort of epigenetics, through sort of these DNA methylation. One of the ways is DNA methylation patterns. The expression, increased expression or decreased expression of these DNA, or well, essentially DNA methylation sort of is kind of these methyl groups that get added on top of these sort of genetic regions, which allow for increased or decreased expression of these particular genes. So think about this, that if your genetic material, if your genes are kind of like all your kitchen sink, or all the materials in your kitchen, right? Everything you need to cook, then your DNA methylation is exactly the recipe you need to know how to make a neuron versus how to make a skin cell. And that's what really DNA methylation is really useful for. But what happens over a period of time is that this recipe tends to fade away. It fades away over time due to environmental factors, due to sort of replication, so on and so forth. And we can quantify how much this recipe is fading away using machine learning algorithms. And that, in the basic sense of it, is what a epigenetic clock is. And we'll talk about this in a minute, but the thing is that these changes in DNA methylation was something we knew for a very long time. We knew that changes in DNA methylation were correlated with age and age-related diseases way back in 1999. But kind of the first ever predictor of chronological age was sort of tried to be attempted to build in 2011, using sort of just 2 CpGs. And then, of course, Steve Horvath came up with his multi-tissue clock, which allowed us to sort of predict chronological age using multiple different tissues. And then, of course, we had sort of the rise of a bunch of different epigenetic clocks. And the basic model of them was that you take a bunch of DNA methylation sites and you try to predict some kind of aging phenotype. And that is potentially sort of chronological age, the rate of aging, mortality, so on and so forth. So So something very simple. It's a very basic machine learning model. And that essentially has been the evolution of these epigenetic clocks in some ways. So you have had the first generation of epigenetic clocks that predicted chronological age, which were kind of prognostic. The second generation of epigenetic clocks, which was significantly more prognostic than the first generation. And then in 2022, I would say, is when we came up, Albert and Morgan came up with the idea of reliability, essentially. So what happened was that a lot of these Gen 1 and Gen 2 clocks had an issue, which was if you took blood from the same individual and ran sort of DNA methylation analysis twice, you would get two very different values for their sort of biological ages. Essentially, if you had two technical replicates, you would get a value of 55.6 years and 47.2 years, as I showed you here. And that just doesn't make sense. These clocks are not reliable in that context. And so to improve that reliability, Albert and Morgan essentially worked on what we call as sort of gen reliability, or the next generation of clocks, which had significantly more reliability. But here's the thing, we could now potentially use these in clinic, right? We could start taking these to clinic and saying like, hey, let's actually use these in clinic and translating sort of this geroscience from bench to patients, right? We could take a patient who goes to a geroscience medicine clinic, have a clinician measure these aging biomarkers like these epigenetic clocks. Based on these biomarkers, a clinician might suggest to them some longevity interventions. A few months later, the clinician will assess their biomarkers to see the intervention's effects, and the patient will leave the clinic ready to celebrate their sweet 16. But that's not what is happening at all. If that was the case, all of us would be going to longevity clinics and probably looking like Kim Kardashians. But Kim Kardashian doesn't really use a longevity clinic, uses some kind of other therapies altogether. But the point is, this is not what is happening in practice, because there are sort of challenges in this process. The challenges include, for example, at the very first step of measuring biomarkers to quantify aging, we need what we call as insightful aging biomarkers. So till now, all of these biomarkers that we have been talking about have been typically quantifying what we call as whole body aging. What that means is that they summarize aging as a single score. Now, what I mean is that they would take your whole blood DNA methylation and they will tell you if you're 5 years older or 3 years younger. Now, that is helpful, but when you go to a clinician, that's not really that insightful, that's not that actionable. What would a clinician really do with the fact knowing that you're just 5 years older or 3 years younger? They need some more insight than that, like to know maybe what organs of yours are aging faster, what systems of yours are aging faster. And how can we do that? So these clocks in some ways are not really insightful, and we'll talk about how can we make these clocks insightful. Another challenge is if you look at the second step, which is finding sort of effective geroprotective interventions. And we don't quite know which interventions are actually geroprotective. And that's a huge problem, partly because if you look at the typical drug discovery pipeline, you go from screening compounds to potentially filtering these compounds in sort of model organisms, and then finally sort of running them in sort of having sort of clinical trials. But when you think about this in the context of gerotherapeutics, the timeline for this looks very different. If you're running these in sort of compounds and screens in cell lines, that's hours to days. In model organisms, that might be days to maybe a few years in mice. But when you're thinking about running these trials in humans, these trials would take decades. The reason it would take decades is because the end outcome here is mortality. You're really looking at how people will potentially die of chronic diseases, how they might actually die, and you're really looking at life extension or human health span extension altogether. And because of this reason, we have almost 1,000+ compounds that are sort of ready to be tested in preclinical stages with which we have shown to extend either lifespan in sort of worms or in mice, but we have not been able to test them in humans at this point in a structured manner, because we just don't have a mechanism of running a clinical trial for up to 10 years. And to this point, what we can have a solution to this is a surrogate biomarker, a surrogate aging biomarker that can essentially be prognostic and responsive. And that is, again, something that we can potentially work on. And then the third challenge here in sort of this clinical translation is when we have this kind of an intervention, we need these biomarkers to be responsive. They need to respond to an intervention. What that means is that when you treat an individual, this particular biomarker needs to potentially go down or go up, right? And if they do not, we just don't know if these interventions work. And so if you think about it, all these clocks that we have, they were initially validated for prognosis, and then of course used for prognosis in a lot of different studies. But we think about responsiveness for these clocks. They've been used for prognosis in a lot of different studies, but they have actually not been systematically validated for responsiveness. They have been used for responsiveness in a lot of studies, but they have not been systematically validated for responsiveness, which is a huge problem. And so again, we quite do not know if these clocks are actually responsive and ready to be used in sort of this clinical setting. So in a lot of ways, there are two critical challenges around aging biomarkers that I would be addressing in this thesis, which is around whether we can make insightful aging biomarkers and whether these aging biomarkers are responsive, and around sort of surrogate endpoints. That is potentially something I think the lab in general would be working on as we move forward, and is one of the goals of the lab. And as I said, this is kind of the chapter, these are the kind of chapters of my thesis: building insightful DNA-M aging biomarkers, which is systems age, a single blood DNA methylation test to quantify aging heterogeneity across 11 physiological systems. And then second, are DNA-M aging biomarkers responsive, which is a meta-analysis of 51 longevity interventional studies in humans across 110 different DNA methylation biomarkers. So the first chapter, which is systems age. A single blood DNA methylation test to quantify aging heterogeneity across 11 physiological systems. So I'm first going to talk about how we trained systems age, and then of course how we validated it, and its replication across different cohorts, and as well as how we can discover what we call as aging subtypes and the value of these aging subtypes. And of course, how systems age is now being used in practice. So the first and foremost, how do we go about training systems age? As I initially pointed out, these biomarkers that are there in practice are very unidimensional. They just tell us whether an individual is 5 years older or 3 years younger. But systems age is something really interesting, and this is my first project in Morgan Levine's lab, where we were able to sort of quantify aging in 11 different organs of your body using DNA methylation by predicting sort of biomarker changes in sort of system-specific biomarker changes, and then sort of merge them into sort of a whole body clock. And the way we did this was a very complicated training pipeline, or is it? So let's try to make this really easy, and I promise I'm not going to put you guys to sleep when I do this. I've already seen some sleepy heads. But the first step of this was doing some dimensionality reduction on the system-specific biomarkers. So we had to integrate hard biomarkers, for example, into unified components. So for example, they were like clinical chemistry biomarkers, like homocysteine, but also physical functioning biomarkers, like heart rate, diastolic blood pressure. And then in the next step, we predicted these components using whole blood DNA methylation. And finally, we built a proportional hazard model using the proxies of these DNA methylation components. So now we don't actually require these original sort of biomarkers at all. We are just using the proxies of these components altogether. And this way we can obtain what we call as a heart score. Eventually, we can combine all of these sort of different organ-specific scores into one unified whole body score. And what this really does is systems age essentially allows to provide potentially deeper insights to physicians. So now you can go from saying a whole body with whole body DNA methylation, you can go from not just saying that you're 5 years older, but you can actually say why you might be 5 years older. Potentially the reason you're 5 years older is because your heart is 11 years older or your lungs are 7 years older, right? And that's potentially really a lot more insightful for a physician, something that a physician can act upon. And of course, we have validated system specific scores quite extensively, almost in 8 different cohorts and across 20K plus individuals and 30 plus aging phenotypes. And one of the ways we validated this was to look at system specific score, system specificity. Now this graph looks a little, this sort of slide looks a little loaded, so let me just quite quickly explain this. On the right-hand side of this graph, you would see what is called a forest plot. What this is simple meta-analysis of associations between a particular score, for example, like the heart score, with a particular outcome, like cardiovascular disease, like coronary heart disease. And what we have done is the meta-analysis is between sort of different racial groups across different cohorts. And what we have taken is the Z-score at the bottom of this meta-analysis and sort of scaled it between different organ scores so that we can really compare which of the organ scores or system scores are the most associated with cardiovascular disease. So in this case, for example, when we compare all the cardiovascular disease scores, associations with the cardiovascular disease or coronary heart disease score, we see that the heart score is the most strongly associated, as we would expect. Now, remember, all of this is being all of this data is coming from just one single DNA methylation blood draw, which is the real power of this test. So let's do a couple of these tests here. What we see is, for example, when we look at something like baseline functional phenotypes, we see like with cognitive functional decline, the brain score is most strongly associated. With physical function, it's the musculoskeletal score. With total comorbidities, it's the inflammation score. With lung cancer, it's the lung. With leukemia, it's blood. With coronary heart disease, it's heart, as we just talked about. With diabetes, it's musculoskeletal. With thyroid disease, it's hormone. With arthritis, it's musculoskeletal. And as you can see, these have been tested in about 5,000 individuals, so a fairly large cohort of individuals. We also compare sort of these scores with some of the very well-known clocks, like median pace, DNA grimage, and PC phenoage. And these sort of specific scores, specific to the organ itself, tend to outperform the presently available clock. What that means is not only are you getting specificity that we are aging the most in the heart organ, we are also getting to these sort of associations are better than the very well-known whole body clock as well, which again just goes on to show that specificity is actually better than sort of knowing the overall body association. And then, of course, we replicated these results in multiple cohorts. So these are just some of the cohorts we replicated these results in. So for example, BLSA is another cohort, and we saw that sort of the diabetes results that we saw in our Women's Health Initiative cohorts, or the WHI cohorts, matched exactly with the PLSA cohorts with musculoskeletal strongest, or in coronary heart disease and coronary artery disease, we again saw replication of our results. We also saw, for example, in ADNI, the brain score was the strongest with within Alzheimer's patients. But when we looked at sort of a larger group of patients which had Alzheimer's, MCI, and were sort of generally healthy, it seemed like inflammation and hormone were the sort of driving factors. And when we actually looked at which particular scores were able to really differentiate between different diagnostic categories, we were able to really see that most of the sort of well-known sort of whole body clocks were not quite able to differentiate between the different diagnostic categories, but our system-specific scores like inflammation were able to differentiate with quite a lot of significance. But the most interesting part of this project is, I would say, sort of the ability to discover what we call as aging subtypes, which is I would like to show like a very small example here. And this is kind of what led to this whole sort of arm of this project, which is we looked at these three individuals. These three individuals have the same chronological age, and they have the same overall whole body age. What that means is that if you were to measure sort of their whole body age, they would look exactly the same. And their chronological age, they would look exactly the same. But as you can see from their different organ ages, they look very, very different. And this is what sparked us on a whole trajectory of understanding that maybe if we are aging in very different directions, in very different dimensions. And this is what we are calling as aging subtypes. So in one of our cohorts of about 2,000 individuals, we categorized individuals based on, clustered individuals based on sort of how they are aging. And so we sort of performed essentially a very basic WGCNA clustering based on their sort of their scores, their organ aging scores, their system scores. And we found sort of these 9 different clusters. As you can see there, and these are the kind of the means of these 9 different clusters, and you can see that these systems they're aging in very different systems in these 9 different clusters. What's really interesting is the predisposition to disease in each of these clusters is very different. They have increased or decreased risk of diseases based on how they are aging in each of these clusters, which is really interesting to me. And I'll give you some examples here. I call these 3 subtypes what sort of the aging, the smoking subtypes. All 3 of these groups have, all 3 of these clusters have an increased number of smokers in them, which is why you can see sort of they have increased heart and lung aging. So on the radial axis is what you can see sort of their rate of aging. And what you're really seeing is an increased risk of heart and lung aging, on the radial axis for these guys. But interestingly, only cluster 8 and cluster 9 go on to develop lung cancer. And only cluster 9 actually goes on to develop coronary heart disease. Something we think is very common, right, with aging individuals, with smokers, would be that these people would develop lung cancer and coronary heart disease. But only sort of a subgroup of these individuals, based on sort of these DNA methylation patterns, seem to develop coronary heart disease and lung cancer, which is really interesting, right? That only a subset of these individuals are sort of predisposed to these diseases and not all of them. And it's worth asking why that might be the case. But I would like to point out sort of another group here, which is what I call sort of the cardiovascular disease clusters. So we just looked at cluster 9, which has an increased risk of coronary heart disease, but cluster 3 as well has an increased risk of developing sort of cardiovascular diseases, such as myocardial infarction, coronary heart disease, stroke, so on and so forth. But interestingly, cluster 3 doesn't have as much increased heart aging or lung aging for that matter. Rather, it has increased metabolic and inflammatory aging. So maybe the mechanisms through which that they are developing sort of risk of having cardiovascular disease is completely different from cluster 9 altogether. Now imagine if they go to a clinician, they would probably be treated the exactly same way. 2 people from these groups coming from cluster 3 and cluster 9 would be treated the exact same way. But maybe, just maybe, based on this data, they should not be, right? And that is something we would like folks to ponder upon even more. And so I just want to talk about a little bit about systems age in practice. As Albert mentioned, systems age has been is in the process of being patented and is actually being licensed out as Symphony Age to True Diagnostics. And even though it's actually still in the final stages of review, it's on bioRxiv and has been cited multiple times since 2023, and been used in multiple different studies, multiple different interventional studies. In just a few examples here, where system scores have been shown to be associated in schizophrenia. Janelle has actually been working on showing associations of system scores with with resilience, as well as system scores have been shown to decrease with plasma exchange therapy, as well as certain specific system scores have been shown to decrease with vegan diet as well. So in summary, I would like to say that systems age has been built to predict aging in 11 different systems and organs using a single whole blood DNA methylation test, and provides greater insights into human aging, hopefully. System scores have been shown to be associated with a wide variety of diseases and conditions, and this has been replicated in multiple different cohorts. System scores can potentially capture aging subtypes, a completely new paradigm that can potentially shift our understanding in medicine. And within a short span of time, the neuroscience field has started to use systems age and system scores to generate insights for diseases and interventional effects. And we continue to validate systems age in larger and diverse cohorts that will further our understanding of systems level heterogeneity in aging. With that, I'd like to talk about my chapter 2. I know people are getting a little bored. I promise I'll make this quick. But please, this is 5 years of my work. I really want to talk about it. Okay, so DNAM aging biomarkers are responsive. Insights from 51 longevity interventional studies in humans. Okay, this project is really, really close to my heart because this has been kind of the baby project that we started in lab, and I don't know what to say about this. This is the project that I'm really excited to talk about. So I'm sorry if I go on tangents in this one, if I wasn't going on tangents already. So I'll talk about what all we are going to chat about in this chapter. So we will first talk about building a database and why we actually need to build a database for sort of all of these longevity interventions and all of these clinical trials. Why is the need, and how did we go about building this? Because that in itself is a huge, huge effort. And then asking the question, do we actually have an interventional database? Like, we can say we have a longevity interventions database, but there's never been actually a longevity intervention. So how do we really know we have a longevity interventions database? And then based on that, asking the question, which DNAM biomarkers are actually responsive to these longevity interventions? And then, of course, which clinical covariates affect responsiveness? And then finally asking, can we go beyond understanding sort of this whole body responsiveness? Sort of my favorite topic about going beyond just whole body responsiveness. So building a database of 50 plus longevity interventions. Decline. Okay, so the first question is, are DNAM biomarkers responsive? And we have had a lot of trials already using sort of aging biomarkers, as I talked about. So let's talk about this one particular trial in the field, a very famous one called the calorie trial, appropriately named after a calorie restriction study, which showed that one of the very famous clocks called unidinase actually decreased upon sort of usage of sort of a calorie restriction intervention. What it did was it looked at sort of unidinase and a bunch of other clocks in the field, but some very well-recognized clocks. And what it found was that a majority of the clocks did not actually significantly go down. They did not change. These crosses that you see essentially tell you that they did not decrease, which was not really useful in a lot of ways. The black boxes that you see actually were the clocks that were never calculated. Well, that's good for us to know that maybe unidinase is the clock that is responsive, and that's the one we should use. Well, then we look at the umbilical cord plasma study, which was a plasma infusion study used from an extraction from umbilical cord of babies. Sounds a little dystopian, but whatever. The study was nevertheless really exciting because it's one way of potentially extending lifespan and health span. I find it really exciting. This particular study did not use unidinase or a lot of the other clocks, but then showed that GRIN-HB1 was the one which was responsive to this intervention. So is GRIN-HB1 the clock that we should be using for interventional studies? I don't know that, right? And then there are these studies that show a lot of these clocks were measured, but maybe Dunedin pace was not necessarily being responsive in the senolytic graph, right? Or primage one V1 was not being responsive. And we don't really quite understand which of these clocks we should be using. A majority of them are actually sort of black boxed out. They were never calculated. So we don't actually even know if they might have been responsive or if they wouldn't have been responsive at all. So what we really need to do is a very systematic analysis across all of these interventions to understand which of these clocks are responsive and which of these clocks are not. And that is what we ended up doing. We calculated all of the clocks across all of the interventions that we could find. We figured out 50 interventions, actually 51, because that's actually it's an interesting story. I'll tell you a quick short story, and no one knows this. There's a reason why I stopped at 51 interventions, because in Hinduism, 51 is a very auspicious number. And so I stopped at 51 interventions in hopes this paper gets passed. I'm not even kidding. The Hindus in the room would know this. So I stopped at 51 interventions that it'll work out. I was hoping for 101 because that's a more auspicious number. We'll hit 101 hopefully in the future. But anyway, so we looked at 51 interventions and 110 clocks. We should have stopped at 101 clocks. And 51 interventions across lifestyle, across pharmacological, across sort of supplements, medical procedures, and a lot of clocks. So each little box that you see on this heat map is essentially one t-test for one particular clock. And what we are seeing with each of these little boxes is the mean difference of scaled epigenetic age deviations pre and post an intervention. So for each of these interventions, we have pre-intervention and post-intervention DNA methylation data, right? So we can actually test if the epigenetic age is going up or down based on this intervention. So these are all sort of some kind of clinical trial interventions, right? So let's talk about how we went about building this, because this was difficult. The first step of this was actually searching through public and private repositories. And even though sort of Daniel and I were leading the charge on this, this was a huge, huge effort with a lot of people in the lab working on this. Pretty much everyone in the lab contributed to this. And as you can see from this sort of picture at the top right-hand corner, we used to sort of sit together in these coding weeks and do this work together. And we, of course, had help from outside, from our collaborators at True Diagnostic, who sort of sent us more data. And we did a lot of this sort of data correlation where we could annotate these data sets and clean them up. And then the main criteria for these data sets that we had was that they should be longitudinal, that they should have pre and post DNA methylation data based on some interventional study, for example, taking metformin for 4 weeks, and it should be whole blood DNA methylation. And then we also curated data for what we call as aging events, such as having COVID or control data, so on and so forth. And of course, Daniel was a huge help in this. And then we calculated lots of DNA methylation clocks and biomarkers. I want to thank, of course, Kira, who was the initial creator of the methyl cipher package, and Jess, who has been the one who has been sort of leading on development of the methyl cipher package ever since, and has been sort of critical in developing this further. And then finally, we have sort of performed paired t-tests on all of these different clocks and interventions. And that gave us this big heat map. So our first question was that, do we have a longevity interventions database? Because as I pointed out, that to answer our question that we have curated a longevity interventions database, we essentially went out and sort of collected all of these interventions based on our a priori knowledge of which interventions might potentially slow down aging, like rapamycin, which has slowed down aging in mice, or metformin, which in retrospective has shown to decrease or increase lifespan in sort of studies, and so on and so forth, or diets or calorie restrictions. So, but there's no sort of one sort of truth here in this context that which of these studies are, or which of these interventions are actually the correct ones. So what we did was we can do like a quick test in some ways to see if a majority of our interventions in our curated database generally sort of decrease epigenetic age across a multitude of our well-known epigenetic clocks. So it's kind of like a crude test of figuring out, are these interventions generally decreasing epigenetic age based on some of the most well-known clocks out there. And so I understand it's kind of a chicken and egg problem, but we have to start somewhere, right? And this is where we are starting. So we took some of the most well-known clocks that are out there, 15 of the most well-known clocks from generation 1 all the way to the latest generation clocks. And what we first did was a very basic check. We compared aging events to controls to all our longevity interventions. So we took all the 15 clocks across 51 interventions, and we plotted them right here. And then we had controls in similar ways, and we had aging events in similar ways. And we saw a very simple trend that when you had an aging event like COVID or chemotherapy, epigenetic age went up. When you had a control, epigenetic age did not change. And when you had an intervention, epigenetic age went down. So in general, to some extent, we can say we did compile a longevity interventions database, right? But let's break this down further. When we break apart these interventions into some categories of interventions broadly, like lifestyle, supplement, pharmacological, medical procedures, we see that broadly across all of those 4 categories, there seems to be some kind of a decrease in epigenetic age, some more than others, of course, right? But broadly, we are seeing decrease in all 4 categories. And then, of course, when we look at all our interventions across the board, we see some interventions are, of course, increasing epigenetic age. Some are, but most of them seem to be sort of decreasing epigenetic age. And so each dot that you're seeing here is the effect size of one whole body clock for an intervention, right? So it's a one t-test for one particular clock. And so you have 15 dots per intervention that you're seeing. And then we have performed a one-way t-test to essentially see how much this particular intervention is deviating from 0 effect size across these 15 clocks. And what we see is that 68% of these interventions show some kind of decrease in epigenetic age, and overall 37% are showing sort of significant decrease in epigenetic age, which in our opinion gives us pretty sort of strong this thing that we have some kind of an interventions database. So it appears we have a longevity interventions database. Now our hypothesis is that if a DNAM aging biomarker is actually responsive, it should be decreasing in effect size when looked across the board for all 51 interventions. So the question now is, which DNA methylation aging biomarkers are responsive? So let me just remind you again about our different versions of clocks. We had the Gen 1 clocks, which were chronological age predictors, the Gen 2 clocks, or 2+ clocks, which were mortality predictors and pace of aging predictors. And then we had the Gen reliability clocks, which were sort of the reliable versions of the Gen 1 and Gen 2 clocks. So we had sort of Gen reliability, Gen 1 reliability clocks and Gen 2 reliability clocks. So again, we have these 15 clocks, and we're just going to actually invert our analysis. So till now, we were compressing our accordion this way. Now we are going to compress our accordion this way. So now, essentially, every dot that you see here on the x-axis, you have sort of our 15 different clocks, and every dot that you have here is one intervention. So the effect size of one intervention, so one t-test, essentially, right? And what we see here is something really, really interesting. We see that overall, these reliable Gen 2+ clocks, these mortality and pace of aging predictors that are reliable, are the ones that are significantly decreasing epigenetic age across the board. And that is really interesting. So there needs to be 2 criterion. One, that you need to be a mortality or piece of aging predictor. And second, that you also need to be reliable. Not one, but all, you have to match both of these criterion to actually be able to be responsive to interventions in general, based on this 51 interventional data. And so the next question we asked was, are there clinical covariates in our data that are actually causing responsiveness or causing more responsiveness? And so we had a lot of clinical covariates across these interventions that were varying, like the average age in these trials, sort of the number of subjects, the standard deviation of the subjects, the intervention period, person female, and the study population, whether it was healthy or diseased. And we saw that the particular sort of clinical covariate that had the most effect was on responsiveness was the study population, whether the group was healthy or diseased, whether the trial population was healthy or diseased. So if the population was diseased already, it seemed like there was a much stronger response as opposed to the healthy population. So as you can see here, if you see something like PC phenoage, it had a significantly stronger response in the disease population as compared to the healthy population. Similarly, in systems age, PC Grimage, PC Hannum, Grimage V2, except for in Dunedin case, which there were almost similar responses, and we'll talk about this in a second. And actually, if you look at sort of these between healthy and diseased groups, what you see is that in general, there are a lot stronger responses in the disease groups as opposed to sort of in the healthy population, potentially pointing out at the fact that, and again, this is just a hypothesis, that diseased individuals are potentially a lot worse off in terms of epigenetic age, so there's more scope for improvement. And again, this hypothesis needs to be tested, and that could be just one of the explanations to this hypothesis. But there's something interesting here, which is Dunedin pace, which is unlike the other clocks, which are all sort of mortality predictors that predict how far away you're from death or mortality. Dunedin pace essentially predicts the rate at which you're aging, which is a slightly different metric, so to speak of. And interestingly, the slightly different metric is potentially able to measure responsiveness in a different manner, in the context that in a slightly unbiased manner, where it's responding quite similarly in both healthy and diseased populations in a relatively unbiased manner. We have done some more work on this as well to understand that whether we can replicate this in sort of other sort of clocks, and we see that there's a potential for replicating sort of this rate of aging metric as well, and the responsiveness sort of remains unbiased. But then our last question is, can we go deeper than just full body responsiveness to interventions? And so till now, we talked about sort of systems age, which can sort of quantify aging in different organs of your body. But there's something else as well that has been working on in the field where we can sort of look at what we call as sort of these biomarker proxies. We can look at sort of specific proteins and metabolites using sort of whole blood DNA methylation. And specifically what you can do is imagine you have whole blood DNA methylation. You can go and figure out if you're 5 years older or 3 years younger, but also then go your heart and lung aging are really high. And then the biomarker proxies can further tell you about, oh, probably your heart is really older, and maybe the reason it's really older is because homocysteine or cholesterol or troponin is really high. Again, these are potential applications. I'm not saying this is quite there yet. But again, this is data just acquired through whole blood DNA methylation. You do not need to do proteomics or anything else. And so what we were able to do is test out about 90 of these proxies, at least in this paper, and Daniel has been able to test about 1,600 of them now. So we were able to test about 90 of these proxies in this paper and saw many of them being responsive, along with a lot of the system scores being responsive as well to interventions. And this is really interesting because I'll give you an example here. which is look at these diets that we have. So we have a lot of diets like low carb diet, low fat diet, green Mediterranean diet, healthy guidelines diet, calorie restriction, vegan diet. And what we can see is across the board, something like musculoskeletal, metabolic, and inflammation, all of these sort of organ-specific scores as measured by systems age seem to be going down with these diets, right? And then additionally, we also see a lot of these sort of protein and metabolite proxies, right? Some of them from Grimmage, some of them from so omic MH. All of them also seem to be showing similar trends, such as sort of the glucose MRS score, HbA1c, DNAM log A1c, which again goes on to show the potential value of these proxies in being used in this context. And I want to talk about sort of a really sort of set of interesting stories here, which is really powerful in my opinion. So if you look at these three interventions are showing decrease in epigenetic age for whole body clocks as we see. So intramuscular plasma concentrate. So this is kind of the umbilical cord plasma study, but it was applied intramuscularly. There's metformin, which you might have heard as the diabetes therapeutics, and there's hyperbaric oxygen therapy. So if you look at sort of the whole body scores, we see that they are decreasing some or the other sort of second generation reliable clock, as we can see, right? But when you look at the system scores, we get a completely different story, right? So for example, the intramuscular plasma concentrate specifically tends to decrease the musculoskeletal system, right? So it seems to be decreasing musculoskeletal aging because it was potentially applied intramuscularly, right? But it actually increases hormonal aging, which is really interesting to me, potentially because it was taken from a very different hormonal system, which is an umbilical cord taken, given to a human. Again, that's just a hypothesis. We probably need to test this more. Metformin, on the other hand, seems to have a much more systemic effect. It seems to be decreasing epigenetic age in different systems, such as brain, blood, inflammation, heart, kidney, metabolic, musculoskeletal, so on and so forth. So it has much more of a systemic effect compared to the other intervention. While hyperbaric oxygen therapy seems to have a very specific effect on lung aging itself. So again, these three interventions seem to have very different effects altogether, something you would not have been able to parse out just by looking at the whole body aging clocks themselves. So in summary, I want to talk about here is that we curated a longevity interventions database. We revealed that Gen 2+ reliable clocks are most responsive to interventions. We also showed that study population, healthy versus disease, has an influence on responsiveness. We observed that unique and pace, a rate of aging metric, uniquely captures responsiveness. And we highlighted the potential of insightful DNA-M aging biomarkers in revealing how interventions may specifically impact humans. So what's next for this project? Well, we're looking at more interventions, potentially growing this to 101 interventions. More DNA-M aging biomarkers. As we pointed out, Daniel is already working on adding 1,600 more. We already have another 1,600 waiting for us in the Atriums project, so hopefully that too soon. We're looking at doing this cross-species, which is really exciting as well, and translate. So let me actually talk about translate, which is really, really exciting as a follow-up. So a lot of challenges await for us in translating sort of some of these things that we talked about to clinics, especially when we think about sort of translating these to these biomarkers as surrogate endpoints in clinical trials. And this is kind of what led to the formation of the Translate project, and has been a spin-out of the Responsiveness project. Initially, when we were thinking about the Responsiveness project, I remember Albert and I, we were just thinking about this from like the perspective of sort of just responsiveness itself. And then we came up with like, maybe there's more to it. Can we sort of standardize these biomarkers from more perspectives? Can we look at prognosis? Can we look at reliability so that we can really standardize these biomarkers and really tell a story around which of these biomarkers are potentially ready for surrogate endpoint status, or at least validation of surrogate endpoint status. And that is why we thought about, can we validate these biomarkers in a more structured format? So for example, for prognosis, we're doing translate prognostic, looking at more diverse and standardized populations. So in typically these biomarkers, when they have been validated, they've been validated in very specific Eurocentric populations or Western-centric populations. So we're trying to look at more diverse populations now, also in very specific aging phenotypes as well. And again, in a few standardized metrics only. I'll skip over this one for now. And then translate reliability as well. We have been looking at not just technical reliability, something that Albert worked on, but also something called as biological reliability, where how does a biomarker change over, these biomarkers change over a period of time, over during the time of day, before and after a meal, because of stress. Because ideally, these biomarkers should not change too much. Because of these different perturbations, as if you want to translate them to clinical trials or want them to be responsive. And actually, in some of our preliminary analysis, we saw that some of these biomarkers may not be as biologically reliable. So on the x-axis, y-axis, we have biological reliability. And we can see that some of these might not be as biologically reliable. Again, this is preliminary analysis, and Daniel and I are still working on sort of improving this. But here's the punchline. I'm not going, this whole talk can be, I can talk about translate for another 50 minutes, and I don't want to bore you guys. But the punchline here is all of these metrics, if we look at all of these metrics, they do not correlate with each other. They do not correlate with each other. And we're just looking at responsiveness, prognostic, technical, and biological reliability. Actually, some of them have negative correlations. And that is a problem, because we have only been optimizing for prognosis. We have only been optimizing for how good a biomarker can predict future disease outcomes, future mortality, not been focusing on sort of biological reliability or their responsiveness. And we need to be focusing on these factors if we need to translate these biomarkers to clinical trials, and that is critical. And that's why this project is so critical. And yes, as I said, prognosis and responsiveness does not equate surrogate biomarker status, but hopefully translate will provide us with a short list of biomarkers that can then be used in a small clinical trial to follow up to see if these biomarker changes are actually predictive of differential health outcomes. And that's hopefully our goal for this project. So in conclusion, many challenges exist in translating geroscience to clinic, but we're working on building insightful aging biomarkers as well as responsive aging biomarkers. And to some extent, we've also been able to standardize interventional comparison using DNA methylation clocks. Of course, we don't know sort of the surrogate endpoint status and whether these biomarkers are ready for that effect or not. But of course, we're working more on sort of getting these biomarkers to surrogate endpoint status using our translate work, which we'll continue to do. And with that, I'd like to do my acknowledgements. I know we're already over time, as usual, with all my presentations, as my lab knows. I'm infamous in my lab for going over time. But I'll take my minutes with my acknowledgements. So bear with me, because this is the most important part. So of course, this is the more official acknowledgements. I want to thank everyone in the Higginson lab, my committee members, Morgan and Rajita, all my collaborators, my funding, the Gruber Foundation, NIH, True Diagnostics, who help with so many things, Impedus grants that sort of sailed me through this PhD in a lot of ways. And one other person who's probably online, Berika, who's been helping me out with a lot of my social media and my personal stuff, personal management, and super helpful. Thank you so much, Berika. And a lot of the scientists who made their data and code available, this allowed the Translate project and the responsiveness project to be possible. Without them, this project will not have been possible. So I really want to thank them. Many of them actually would not even know about me. They just made their data and code publicly available. But as As a computational biologist, it is so easy to sit in my silo and use someone else's data that has been generated and put on Geo and not thank them. So I really want to be grateful for them for working, doing the good deed and putting up good data so that we computational biologists can sit on our computers and work remotely and do the easy stuff because you guys do the hard stuff. And I want to now do some personal acknowledgements. So it takes a village to raise someone like me. From two pairs of ever-loving grandparents who, unfortunately, none of them are with me anymore. And short backstory, three of them actually had cancer and potentially what propelled me to study biology in the first place. My paternal grandparents actually, both of them got cancer at a very young age and were discovered, like when the cancer was discovered, it was pretty much metastatic. And why I wanted to study biology in the first place, unfortunately, there was not much scope to study biology back home in India. It was actually frowned upon. The only way to go was studying computer science. That's what good boys do. But I did have a very supporting family. I will tell you that. My parents, who, by the way, I will tell you this. No one in my family in my extended family, has ever studied science, let alone biology. But they were always supportive of me pursuing what I needed to pursue. My parents, my loved ones, and Kaveri, I'll thank you later, but I'll thank my parents first. My mom's an English teacher. She inculcated in me a love for language and communication that has allowed me to really perpetuate all the science that I can perpetuate in the best way possible. And had it not been for her, I don't think I would be able to communicate as well as I can right now. Being South Asian is a double-edged sword sometimes. If you know English well, they tell you you know English too well. And if you don't know too well, they don't listen to you. But it was my mom who taught me to keep my head held high when it comes to language. And she taught me how to speak well. So I thank her for that. My dad, on the other hand, he's a marketing and business personnel. And that is where I get my selling point. That is how I know how to sell things well and sell myself when it comes to the world. And it is his perspective on all of these things, which has really allowed me to thrive in an academic perspective. This perspective of being able to be in science while also being able to translate science, that is something that comes from him. Even though he doesn't understand everything I do, I shit you not, when I first joined computational biology, he used to say, I do information technology, I do IT services. But he does know, when I go to him, he does know how to give very good advice on how to translate the kind of work that I do, how to sell myself. So I thank him for that. And Kaveri, thank you for always being there, but we'll come to you in a bit. IIITD was my undergrad college, which no one would know about here, but it was the place that really made me grow. There were two people from there I really want to thank. Nandika, one of my closest friends, who was always there with me as a support, and Professor Raj Iyer, who is no longer here with us now, but he had this booming voice, and he would talk in a room of 180 people without a mic. And he was an ethics professor in an all engineering college. So colleges in India were only for, like, you had an engineering college or you had a medicine college. So being an ethics professor in an engineering college was the oddity. And this man would talk about ethics with the most fervor possible. And he inspired me so much about science and the way to think about science. I cannot thank him enough. So he has to be here. And then Elucidator. My journey to Yale would not have been possible without joining Elucidator. Um, I started at Elucidator when I was in triple IT. This was this was a nothing startup back then. It did not exist back in the day when I started. Actually, the startup I joined was called Gvomics, and it failed. And then my boss from that company started a new company called Elucidator and hired me part-time when I was in college. And then I became the first data scientist, one of the first data scientists, and then went on to lead product management and did that for 4 years before coming to Yale. And AJ and Shweta were on the right-hand side, were the co-founders of this company who supported me so much. These people here are all the team members I had back then. Rajat, one of my closest colleagues there, was a huge mentor. Chef, who might be on call, who actually texted me this morning, one of the smartest people I know, supported me all through the way, my time at Elucidator. And I made some really amazing friends at this place. Raisa, I might remember Raisa. Raisa was one of my closest friends there and still is. So is Nikita. I actually just visited her after so many years. And Pulkit, he's actually getting married this Sunday. And I'm actually, right after my thesis defense, I'm actually flying to India for one night just for his marriage. That's how close we are. So yeah, I made the closest friends at this company. And if there was anything that I can recommend to anyone, always in your lifetime, just for once, go to a startup. It's the best experience you could have. And then Shubhra, my best friend, who I have ranted to, cried, laughed, done. I cannot even tell you the support of my life. She has been with me forever. I met her at Elucidator, and she has been with me till now and will hopefully be with me till the end of my life. She has been the very support under my wings, and I hope she stays like that forever. And when I moved to Yale as a postgrad, I was supported by some really amazing mentors, Becky, Stefan, Dick, and most of all, Graham. Graham, thank you for being here. I really appreciate it. But most of all, I appreciate our quarterly lunches where I come to you for advice, and you have always been so open to give me so much advice, and I really thank you for that. Thank you for being there all of these years. And you know what? You really didn't have to help me, but you did, and thank you. Thank you, Graham. And then again, moving to New Haven was not easy at all, but having all of these friends really made it super easy. And some of them are right here, Gretchen, Kira. It's been a pleasure to have you as my friends. Gretchen, you were one of my first friends in the PhD program. So thank you for being there all through the journey. Really. And my CVB friends, all of you right there, thank you so much again for being here, for being through it all. I really, really appreciate you guys. And again, there was so much support from so many other people along the way. Peter, Anna, Varun, Steve, Jamie Justice, Ryan, Steve Kleinstein, Rajita, Ethan. Many of you are on call right now. I really thank you for your help and your guidance, your advice, for the opportunities you provided. I cannot thank you enough. Very often I even come to you for practical advice, and I really thank you for being there through it all. And mentors, Morgan, I cannot see you right now, but I have to tell you this, that you were the person who really inspired me to switch a field, which I was very adamant about. When I came to Yale, I thought that I'm going to be studying cancer biology. But that one talk from you around aging biology really transformed my perspective on science and biology. And I cannot thank you enough for doing that for me. And the time I spent with you under your mentorship, I learned so significantly. I have cherished that with me till now and will forever cherish that. I have really appreciated every learning from you. And so thank you so much for it. And Albert, I don't have enough words. You have been a teacher, colleague, mentor, psychiatrist, therapist. What role have you not played for me? I still need to Venmo you for our last therapy session. Mm, but we have had the best of times, the worst of times. We have had amazing times. And it has been not just an honor, it's been a privilege and the most fun time to be your mentee. Thank you for having me. And my lab mates. My lab mates, John. I don't know where John is. Oh, he's guarding the food. My God. Well, he can hear me? Okay. Yeah. John, I love you. You have been. John has been John was one of the first people I met at Yale during my PhD program. And he has been a friend, a confidant, a lab mate, a colleague, and has always been by my side through all of these years. So from the person who I went to take advice to join Morgan's lab, to sort of supporting me through all of these years, John has always been there for me. Daniel, we have probably spent the least amount of time in amongst all of these people, but I have become so close to you, I cannot tell you. He's the person who sits right next to me on my desk, and we yap all day, and we probably eat Janelle's head off, and she's sick of us so much. We talk about the darnest of things, like from Interstellar to Dune and whatnot, but I love it. I just love sitting next to him, and I love working with him. He is the smartest person to work with in general. Yaro, I hope you're online, and I cannot tell you, you have been such a support. Working with you is 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 like working with one of the most collaborative people, telling you and giving you a task to work, and you just get it done. I have never worked with someone who works with such efficiency and such clarity, and I wish I wish I get to work with everyone like you, because you're one of the most efficient person to work with, and I really cherish working with you. Yes, you are so effervescent and so energetic. You bring effervescent and energetic. Yeah. Yeah. What? You bring so much energy to a room, and you are so smart. It is a delight to have you. And Janelle, oh my God, you working with you, I could not imagine how smart you were and how quickly you picked up things in the lab. And honestly, every project that I work with you, I am amazed at the pace with which you pick up things and have been able to just sort of honestly take things away from me so that I don't have to work on those things. And you joke about things that I come every flight I go to India and come up with a new project. The reason I'm able to do that is because of people like you who can do these new projects for us. Had I not have people like you in the lab, I don't think I would have been able to think about these new projects. So thank you for being there. Yeah. And last but not least, well, I do have a last but not least actually. um, there are a few people who always stood by me through thick and thin. Kaveri, my sister, who was there for 2 years when I was here at Yale post-COVID, supported me through some of the most difficult times. Who I cried with, laughed with, fought with. I have we have pretty much done everything a pair of siblings can do. And God, I miss you not being here, but I'm so happy that you're in Boston with Sarvanch. You can cry. And my parents, of course, I've already thanked them, but I miss that they are not here today, but they'll be coming for my convocation, which is what I'm excited for. And Joe, thank you for being here for the last 5 years. Without you, this would not have been possible. You were there when no one else was. And you were there through COVID, and you were there through the worst times, through the best times, and I've given you so many headaches. I know I have ruined so many date nights over so much stress. You were there, you stuck by it all. And I promise, I'll make things easier now after this PhD. Things would get better. But thank you. But last but not the least, I want to thank me for all the hard work, for all the late nights, for believing in me, for just being me. Because I could not have done it without myself. So thank you to me. Thank you. Like, do you have like a sword which goes on my... Yeah. I annotate you, doctor.
Officially, I'll say that he's conferred a PhD. Got it.
Thank you.
We're good.
Right.
Congratulations.
Thank you, Rajita. Thank you, Morgan.
Let's more forms.
All the best. Take care. Bye-bye.
Thank you.
Congratulations again, both of you, actually.
Yeah. Congratulations to both.
Yeah.
Thank you.
Bye, guys.
Bye.